Welcome my colleagues. Today I will deliver a brief seminar on phylogenetic relationship of some species of Solanum based on morphological, biochemical and cytological parameters. So you all know about the Solanum. So Solanum is the largest genus among the 98 genera of Solanaceae family, which represents nearly 1% of the total angiosperm flora in the world, and that distributed throughout the world, especially in tropical and warm temperate region. This genus is very much useful for its food value and pharmaceutical demands due to presence of different types of secondary metabolites. This genus has a paradoxical and confusing taxonomy, so it shows much more uniformity in external appearance but exhibited some contradictory external diversified morphology. So there has always been debates regarding identification, phylogenetic relationship and proper taxonomy position among the species of Solanum based on only morphological aspects. So nowadays data from biochemistry, molecular biology, anatomy, cytology, etc. along with the morphological study are used to establish more accurate phylogenetic relationship among the species of Solanum. So these are my selected plant species in my study. So this is Solanum nigram, this is Solanum americanum, this is Solanum violos, this is Solanum xanthocarpum, this is Solanum torvum, this is Solanum macranthum, this is Solanum sicymbipholium, this one indicum. So there are 8 plant species on which I have studied, and among which only this plant is a tree plant. This is called Solanum macranthum. Okay, so the objective of the present study are the morphometric characterization of leaf and seed to assess the external variation of selected species of Solanum. My second objective was cytological characterization to study cytomorphometric variation of selected species of Solanum. My third objective is biochemical characterization to evaluate genetic variation among the species of Solanum, and finally to construct a phylogenetic relationship among the species of Solanum through dendrogram analysis. Now these are the materials and method. Regarding the morphological characters, I have studied the seed length, seed width and dry seed weight, fresh leaf lamina measure, etc. And regarding the cytological aspect, I have studied the number, total genome length, mean chromosome length. And regarding the biochemical character, I have studied the total amount of soluble seed protein, soluble leaf protein, and the total content of chlorophyll. And for the phylogenetic relationship, hierarchical cluster analysis was performed based on above mentioned morphological, biochemical, and cytological parameters using the SPSS computer software. Now these are the results. These are the leaves of my studied plant species. So there are 8 different types of leaves, and their phyllotaxy is alternate. Their margins are more or less wavy. Solanum vilosum and Solanum americanum have slightly serrated leaf lamina, but Solanum xanthocarpum and sicymbipholium have highly serrated leaf lamina. These are the adaxial leaf surfaces, and these are the abaxial leaf surfaces. Okay. The Solanum torvum, macranthum and indicum have sinuate leaf, and leaf apex is more or less acute. The texture of the leaves are soft. In case of Solanum xanthocarpum, Solanum sicymbipholium and Solanum indicum have a hairy glabrous leaf lamina. Regarding the spine morphology, Solanum torvum and Solanum macranthum have spine on petiole, and Solanum xanthocarpum and Solanum sicymbipholium and indicum have pointed spine on both surfaces of the leaf lamina. Now these are the 8 different, these are the seed morphology, and this histogram showing the variation in number of seeds in each fruit among the species of Solanum, and that histogram showing the variation of seed morphology, which contains the seed length, seed width and dry seed weight. And these are the comparative accounts of the seed morphology which are depicted in the histogram. Now these are the somatic chromosomes, that is 2n number of chromosomes of 8 selected plant species, and these are the microscopic views, and based on those microscopic views of karyotype, those karyogram of the 8 selected plant species, and all the species showing the 24 number of chromosomes except Solanum nigram, that plant showing 2n equal to 4x equal to 48 number of chromosomes. And based on those cytomorphometric data, this table is actually designed, and this table showing the somatic chromosome number. These are the somatic chromosome number of 8 plant species. These are the total chromosome length, among which Solanum nigram showing 109.6 micrometer in total chromosome length. This column showing the mean chromosome length. This column showing the total chromosome volume, and this column showing the ratio between longest and shortest chromosome, and these are the karyotype formula. All the plants showing mainly 2 types of chromosome except Solanum americanum. These are showing 4 types of chromosome, and Solanum indicum and Solanum sessimifolium showing 3 different kinds of chromosome. And this is the Pearson correlation analysis based on karyomorphological character of 8 selected plant species, where this is the length of longest by shortest chromosome, this is the mean chromosome length, this is the total chromosome length, and this is the total chromosome volume. And regarding the biochemical analysis, I have studied the 8 plants based on the total soluble protein from leaf, total soluble protein from seed, and total chlorophyll, leaf chlorophyll content. And based on this Solanum indicum showing the highest amount of total soluble protein, Solanum nigram showing the highest amount of total soluble seed protein, and Solanum sessimifolium showing the total highest amount of total chlorophyll content. And based on those data, this dendrogram is actually constructed, and this dendrogram showing the mainly 2 cluster, upper cluster and lower cluster. All the 7 plant except Solanum macranthum are under cluster upper cluster, and upper cluster is divided into 2 subcluster, upper cluster 1 and upper cluster 2. The Solanum nigram complex, that means Solanum nigram, Solanum americanum and Solanum villosum, those are placed together along with the Solanum xanthocarpum, and those are placed in upper cluster 2. And Solanum torbam, indicum and sessimifolium, those 3 plants showing close association based on all the above morphological, cytological and biochemical characters, and those 3 plants are placed together in upper cluster 1. So now the summary. The selected species of Solanum exhibited highly diversified features in each of the concerned morphological, biochemical and cytological parameters. Solanum macranthum exhibited maximum seed length, seed width, dry seed weight, leaf area and fresh leaf weight. Solanum americanum exhibited minimum seed length, seed width and dry seed weight and total amount of soluble seed protein present per gram tissue. The karyomorphology of the Solanum species exhibited gross similarity with some variation. All the species are naturally diploid, that is 2n equal to 24, except Solanum nigram, which is tetraploid. That means 2n equal to 4x equal to 48. Solanum nigram exhibited largest genome size and highest amount of soluble seed protein per gram tissue, and shown lowest fresh leaf weight. Solanum villosum exhibited least leaf area and contained minimum amount of leaf chlorophyll per gram tissue. Solanum nigram exhibited lowest amount of total soluble leaf protein per gram leaf, as well as total content of leaf chlorophyll per gram tissue. Solanum macranthum contains minimum amount of in each gram of seed. In respect of cytomorphometric characterization, Solanum xanthocarpum exhibited smallest genome size as well as mean chromosome length. Longest chromosome is found in Solanum sessimifolium. Solanum macranthum or the giant potato tree exhibited a high range of diversified character in all concerned aspects, mainly seed and leaf morphological features of the species, such as seed length, width and dry seed weight, leaf area and fresh leaf weight exhibit greater percentage of dissimilarity than other species. So therefore the macranthum, that means Solanum macranthum, is placed in a separate cluster in dendrogram. Now I would like to acknowledge Professor Nirmala Banerjee, my teacher and supervisor, for his constant guidance, encouragement and cooperation. Professor Rup Kumar Kaur, Professor Nirmala Banerjee and Professor Kashinath Bhattacharya, the former heads of the Department of Botany, Vishwabharati, for providing me the research facility in this department. I would like to thank my dear fellow lab mates for their constant support and amicable help. I must thank my principal of my present college for his encouragement and support and providing my necessary leaves. I must thank to my colleague Dr. Sondipon Chatterjee and Dr. Chandik Malakar for their constant and moral support. Thanks a lot for patience sharing.